All right, welcome everyone. We're going to be talking about DNA replication in this part of the Biochem Step 1 Thoughts videos. DNA replication occurs in the 5' to 3' direction in a continuous fashion and also in a discontinuous fashion in the other direction, right? So those other discontinuous fashion is going to be compartmentalized into Okazaki fragments. You might have remembered this from college. It's semi-conservative and it's more complex in eukaryotes than in prokaryotes, but they share the same, roughly the same analogous enzymes. So we're going to go through a few details here. The origin of replication, particular consensus sequence in the genome where DNA replication begins. It may be single, such as in the case of prokaryotes, or multiple, such as in the case of eukaryotes. And this is typically what we call a Tata box, like Tata box regions, like AT-rich sequences are found in the promoters and origins of replication. The replication fork is a Y-shaped region around the DNA template where leading and lagging strands are synthesized. You'll see the you'll usually see the helicase in this area. The helicase unwinds the DNA template at the replication fork, and it's usually, and you can see in like Bloom syndrome. It might be deficient in that syndrome, which is, by the way, a BLM gene mutation. Single-stranded binding proteins prevent strands from reannealing or degrading by nucleases. DNA topoisomerases create a single, such as in the case of topoisomerase 1, or a double, such as in the case of topoisomerase 2, stranded break in the helix to add or remove supercoils as needed due to underwinding or overwinding of DNA. So in eukaryotes, you will see irinotecan or topotecan inhibit topoisomerase 1, etoposide or teniposide inhibit topoisomerase 2. In prokaryotes, you'll see fluoroquinolones inhibiting topoisomerase 2 or DNA gyrase and topoisomerase 4. Primase makes an RNA primer on which DNA polymerase 3 can initiate replication. DNA polymerase 3 is usually seen in prokaryotes only. It elongates the leading strand by adding deoxynucleotides to the 3' end, and it also elongates lagging strand until it reaches the primer of the preceding fragment. DNA polymerase 3 has a 5' to 3' synthesis and proofreads 3' to 5' exonuclease. Some of the drugs block DNA replication also have a modified 3' OH group. Thereby, they prevent addition of the next nucleotide, which is also what we call chain termination. DNA polymerase 1, you'll see this in prokaryotes only. It degrades the RNA primer, replacing it with DNA. Same function as DNA polymerase 3, but it also excises RNA primer with the 5' to 3' exonuclease. And DNA ligase catalyzes the formation of a phosphodiester bond within a strand of double-stranded DNA. So one thing you can think of, it joins Okazaki fragments together. Telomerase is seen in eukaryotes only. It's a reverse transcriptase, which is RNA-dependent DNA polymerase that adds DNA, specifically the TTAGGG, to the 3' ends of the chromosomes to avoid loss of genetic material with every duplication. It's usually upregulated in progenitor cells, and also it's often in cancer, downregulated in aging and progeria. Telomerase is going to be very high yield here. You know, you'll see that a lot of people talk about telomeres in aging and cancer. It's a very hot topic. In fact, a lot of people who are trying to come up with the magical pill to live forever are wondering about telomeres and seeing if, because you know, as you progress, as you age, because of the nature of DNA, it gets shorter and shorter, right? So that whole telomere thing is going to be you might see that as a very interesting thing in the future. So just a few things to nitpick. If you don't know about what DNA looks like, go on Google Images and look it up. If you don't know anything about these, like leading strand, the origin of replication, the lagging strand, the primase, you know, how the RNA primer and the DNA polymerase 3, I would take the time to like look through and just see a visual of these things because there's very high yield. And I hope you enjoy this video.